right, welcome. It is my great pleasure to introduce Dr. Wilfried Pastor, who got his MD PhD from Columbia, where he studied the effect of the mevalonate pathway on p53 mutant cancers and appreciating the role of mutant p53 rather than just loss of p53 and how that affects tumor cell biology. He then came and did his residency at MGH and his fellowship here. He did a postdoctoral fellowship with Tyler Jacks and Brian Walpin, and really looking at how to study the T cell response to pancreas cancer. Made several important mouse models where he put antigens that we could track into the pancreatic tumors and looking at how T cell responses evolved and how they could be de-inhibited with things like PD-1 blockade, but also blockade of TIGIT, which is another co-inhibitory molecule and perhaps a little more important in pancreas cancer. So he published that work in Cancer Cell and then has really followed up in humans. And I do want to applaud Will for doing this and saying, if we find something in mice, is this actually important in humans? And so he managed to get funding from the Lustgarten Foundation to run a maintenance trial in patients with pancreatic cancer of TIGIT and PD-1 co-blockade with agonistic anti-CD40. So we'll stay tuned how that is playing out. He came back to Dana-Farber as faculty in 2023 and has really been a key member of our Hale Center for Pancreatic Cancer Research, as well as our collaborative efforts with Breakthrough Cancer. He has some exciting stories continuing to work on T cell responses in pancreatic cancer, which we will hear about today. So thank you.
Awesome. Well, Stephanie, thank you very much for the I'm going to turn this one off, very kind introduction. So I'm going to step away from the podium. Tell me, hopefully you guys can hear me. Really excited to talk today about a story we've been working on, which is not mouse models at all. It actually started out as a antigen discovery effort in pancreas cancer, but work that's really centered on the role of non-canonical or cryptic antigens in this disease. Now, these are my disclosures, none of which are even remotely relevant at all for today. So starting off, though, I wanted to talk about the anti-tumor immune response in pancreas cancer. Now, for a long time, it's been thought that the immune system just can't recognize pancreas cancer, and this is because pancreas cancer is notoriously recalcitrant to immune checkpoint inhibitors, importantly PD-1, PD-L1, CTLA-4 combination, although we hope maybe some of these novel checkpoints may change that. This has been attributed largely to two facts, which are irrefutable facts, in fact. That pancreas cancer has a low to intermediate mutational burden, and so for a long time it had been thought that there are just a lack of neoantigens for the immune system to recognize. And then also pancreas cancer harbors this profoundly immune suppressive microenvironment. Counter to that, though, work from my own lab and others have shown that a subset of pancreas cancer patients do in fact harbor at least predicted high affinity mutation-derived neoepitopes. Elegant work from multiple different groups over the last year have demonstrated that neoantigen-directed vaccines can in fact elicit T cell responses in this disease, although the full clinical outcomes are still being determined. And while anecdotal, there's some really exciting reports showing that neoantigen-directed TCR-engineered T cells, or TCR-T therapy, can actually elicit really robust and durable clinical responses in human pancreas cancer, including in metastatic or advanced pancreas cancer. So how can we leverage the anti-tumor immune response? For me, I think it's sometimes helpful to take kind of a step back and take a 10,000-foot view. This is the so-called cancer immunity cycle. Many of you have seen this before, but I do think it's one helpful way to really think about what are the necessary steps really required to mount an effective anti-tumor immune response just to cancer, not just to pancreas cancer. Now, of course, there need to be antigens for the immune system to recognize. Professional antigen-presenting cells, dendritic cells, and others need to be able to survey the tumor immune microenvironment, uptake those antigens, traffic to a lymph node where they can actually prime and co-stimulate T cells. Those T cells, once primed, can actually traffic throughout the whole body. They need to be able to extravasate, infiltrate into the tumor, where they need to be able to recognize their cognate peptide MHC, or at least for human, peptide HLA, and mediate tumor cell killing. The reason I like thinking about this is we can think about for different cancer types where this cycle might break down. And this slide is really trying to summarize about a decade of work all in one slide, but really showing that pancreas cancer can break down at almost every step along this path. I already mentioned a lot of the lack of response to immune checkpoint inhibition has been attributed to the low mutational burden in this disease, suggesting that perhaps there are just, again, no antigens for the immune system to recognize. Really elegant work from Dave DiNardo, Bob Vonderheide, Hans Schreiber, and others have demonstrated that in pancreas cancer, and in addition some other tumor types, that there's really a paucity of professional antigen-presenting cells that are able to actually take up and present those antigens, effectively leading to a situation where you have suboptimal or even complete lack of priming a T cell response. And if you don't prime a T cell response, you can't reinvigorate that with PD-1 blockade. Even for pancreas cancer, though, there is a subset that do prime a T cell response, and there's really good evidence to say that the immune microenvironment is profoundly immune suppressive, can both passively and actively exclude CD8 T cells. For those that are able to get in, they can quickly be rendered dysfunctional. And there are also evidence to say that tumor cells might become intrinsically resistant to T cell-based killing. Now, the other reason I like breaking it up like this is because you can think about where these obstacles lie might guide how we think about overcoming these. And so cellular therapies, and when I talk about cellular therapies in this talk, I'm going to talk specifically about TCR, engineered T cells, so TCRT. But cellular therapies, T cell engagers, as well as therapeutic vaccination can really overcome or completely bypass defects in that first half of the cancer immunity cycle. And cellular therapies can be further engineered to overcome some of the defects in the second half as well. However, if we're going to really develop effective antigen-directed therapies, we actually have to know what the true antigen landscape in this disease looks like. And although there have been many efforts to computationally predict what the antigen landscape is, what we wanted to do is profile that directly. So what we've been doing actually, and this is work that started about 3 years ago, is performing high-resolution proteogenomics on pancreas cancer using patient-derived organoids. So why organoids? So organoids are really, really important here because this is a H&E of pancreatic ductal adenocarcinoma, or PDAC for short. This is the most common subtype of pancreas cancer. And here you see these islands of malignant cells. These are the cancer nests kind of embedded in this ocean of desmoplastic stroma. That stroma is made up of non-malignant cells, importantly. So pancreas cancer, unlike many other solid tumors, only about 10, 20, sometimes 30% of the overall cellular mass are actually made up of malignant cells. So what we've been doing is using patient-derived organoids or organotypic culture as a way to purify out malignant cells, expand biomass, prior to performing whole genome sequencing, RNA sequencing, HLA typing, and mutation calls, all followed by immunoprecipitation of HLA class 1, elution of peptides, followed by mass spec, i.e. immunopeptidomics. This actually allows us to directly identify, not so much predict, but directly identify what peptides are actually being presented by HLA class 1 in these tumor cells. And I'll highlight while this slide's still on, this has been a big collaborative effort by many folks in the Hale Center who have helped generate these organoids, most notably Andy Aguirre and Sri Raghavan. And all of the mass spec work has been done with the CAR group over at the Broad Institute. So what do we actually see? First, we get great depth. I mean, this is all the CAR group really, but we're finding anywhere between 8,000 to over 18,000 unique class 1 associated peptides per patient sample. And this sounds good, but maybe just to put this in context, this is comparing this to the only prior immunopeptidomic work in pancreas cancer that often found only a couple hundred peptides per patient sample. Again, I'd love to take credit for this and say that this is organoid culture, but it's really not. This is really the mass spec guru nature of the CAR group over at the Broad. However, organoids do, beyond just depth, they actually do add a lot here. And this is with the specificity. So we had hypothesized, right, is if a bulk tumor is only about 20 to 30% malignant cells, we had hypothesized that if you just ground it up, did immunoprecipitation of class 1, eluted those peptides off, many of the peptides you'd find would be coming from non-malignant cells. But we wanted to prove it to ourselves, and so what we did is we took 11 patient-derived organoid lines here on the top and 3 unmatched bulk tumor specimens straight from Whipple resection, did parallel immunopeptidomics. We take all of the peptides we identify, map them back to their parent source gene, and then overlay these onto single-cell RNA-seq data sets. Really nicely with what we had hypothesized here is in the bulk tumors, we can see not just some, but the majority of peptides we're identifying are actually coming from non-malignant cells. These are being contributed by cancer-associated fibroblasts, macrophages, and other cells within the microenvironment. Whereas obviously, organoids are a near pure malignant population, so we can be very confident that these are actually tumor-derived peptides being bound by HLA class 1. So because we had done whole genome sequencing, HLA calls, mutation calls, we could create a personalized proteomic search space for each patient. And we first wanted to look and say, well, in pancreas cancer, can we actually detect evidence for these mutation-derived neoepitopes? And we were actually really excited. We could empirically identify a handful of mutation-derived neoepitopes endogenously transcribed, translated, processed, and presented on the surface of tumor cells. This was very good and actually got us happy that this fit with our computational predictions. However, it was a bit sobering because if you look at the number of exonic mutations actually present by whole genome sequencing, compared to the number that we detect on the surface of tumor cells, it was a minuscule fraction. And I will say, this fits with a lot of other immunopeptidomic efforts in even higher mutational burden cancers, suggesting that only a small fraction of the number of mutations present at the DNA level are actually expressed, processed, and presented truly to be detected by mass spec technologies on the surface of tumor cells. I want to emphasize the kind of every piece of that sentence, because again, there may be some that are actually being presented at levels lower than our detection limit. But what we could directly identify despite really high depth profiling was only a handful of mutation-derived neoepitopes. So this led us to ask, well, all right, beyond mutations, what could we look for? How could we actually target these cells? And so elegant work from Kathy Wu, Aviv Regev, Ellie Van Allen, and others really over the last 4 or 5 years has demonstrated that cancer cells wind up going haywire, right? We kind of know that. We know they start turning on cancer testis antigens, oncofetal antigens, but it turns out that they can turn on novel splice variants, inappropriately retain introns that can be translated, turn on endogenous retroviruses, translate circular RNAs, turn on new post-translational modifications. But importantly, not just the transcriptional machinery goes haywire, but the translational machinery also goes haywire. These are these so-called new ORFs, or novel unannotated open reading frames, been well described in other tumor types, that cancer cells can wind up translating areas outside of the annotated protein coding genome. These are 5' and 3' UTRs, translation in the wrong frame, so kind of the +1, +2 frame, or what are called int-orfs. Translation of long non-coding RNAs or pseudogenes, turns out they actually have coding potential if you mess with the translation machinery. So we wanted to look for these in our tumor cells, and so here, because mass spec is not sequencing, you have to augment the proteomic search space to see them. So here we're looking for new ORF-derived peptides, which were informed by ribosome sequencing to look at active translation, as well as retained introns, which were called using multiple different computational pipelines. Unlike mutations, now we can detect these non-canonical HLA-1-bound peptides in every patient sample, making up anywhere between about 2 and 8% of the overall immunopeptidome. These fell into a number of different defined categories. Again, translation of the 5' UTR, translation out of frame, and again, I want to maybe emphasize here that out of frame or these int-orfs are not due to a frame-shifting mutation, but are actually aberrant translation in the wrong frame. If we aggregate these all together, we can again see that translation from the 5' UTR is the most frequent, but ultimately, again, translation in the wrong frame, frequently observed in these tumor cells as well. We did a ton of work trying to figure out, well, are there differences between these non-canonical peptides and their canonical counterparts? And I'll suffice it to say there's not many differences. I mean, if you look at kind of the peptide length, there's all class 1 associated peptides, so these are kind of 8 to 11 mers. If you look at their predicted peptide motif for different HLAs, again, no big differences here. Some subtle differences in terms of where a non-canonical peptide tends to arise from its parent open reading frame, a little bit more C-terminal compared to canonical peptides, as well as some interesting preference for binding certain HLA allotypes over others. But again, by and large, these aren't very distinguishable between them. We also wanted to look just and ask the question, well, are these all just a product of organoid culture? I mean, it's a very artificial system where we're embedding in a 3-dimensional extracellular matrix, giving it a ton of growth factors. And although we had lower depth of profiling from bulk tumors, we wanted to see if we could still detect them. So we went back to those 3 bulk tumors that we had profiled, again, augmented that proteomic search space to look for these, and we could find non-canonical HLA-1-bound peptides direct in situ from bulk tumors as well. Again, a lower fraction, as one would guess, because these are not all malignant cells, but making up anywhere between 1.5 and about 4% of the immunopeptidome. These fell into almost identical kind of biotypes or categories, again, with a preponderance from the 5' UTR, as well as translation in these alternative reading frames. But this is all kind of just looking at a big level. What about individual peptides? These are largely not mutated, so we can say, are these actually shared amongst patient samples? So we wanted to look between different organoids as well as different bulk tumors. The bulk tumors here are indicated in red, each of the organoids in black, and we're looking at evidence that the same non-canonical open reading frame or ORF are actually translated across multiple patient specimens. And here we could find a high degree of sharing between patient specimens, both between organoid to organoid, bulk tumor to bulk tumor, but importantly, bulk tumor to organoid as well. So we were excited. We said, all right, we figured out that these non-canonical peptides are actually presented in pancreas cancer. This is exciting. But then we kind of took a step back and we're like, all right, well, actually, these are well described in other solid tumors, other hematologic malignancies. So if anyone had just looked for these by mass spec, they would have found them. So instead, we said, well, what are the real open questions in this field? Not just in pancreas, but what don't we know about these non-canonical peptides? Many papers still, reviews and even other papers, will just blanket call these neoantigens. They may be, but they're not mutated. They're not from a virus. So again, I think we ought to have a really high bar for calling something a tumor-specific antigen or a neoantigen. So I think the jury's still out on how truly tumor-specific they are. why are these being translated? What's the underlying mechanism that's governing this non-canonical translation in cancer cells? Are these just coming from stochastic errors in translation? Do these actually encode functional microproteins? Are these actually just coming on as a byproduct of oncogenic transformation? Again, neoantigen implies that they can provoke a T cell response. If they're foreign, sure. We don't know that. And again, outside of some anecdotal reports, we don't really know how immunogenic these are. Can they truly be recognized by CD8 T cells? And lastly, if they can, what's the therapeutic potential? Can we actually use these to guide therapies? So we felt that these were kind of the big four unanswered questions in this field, and so we tried to answer each one in line, and I'll try to hit each one of them in the remainder of the talk. So the first is tumor specificity. Because these novel unannotated open reading frames, or NUORFs, are thought to be aberrantly translated in cancer, we felt pretty strongly that transcription-based approaches, RNA-seq, was not going to be sufficient to do this. So for this, we developed a translation-centric filtering pipeline to assess for translation of these NUORFs in healthy tissues. Here we're leveraging hundreds of healthy tissues profiled by class 1 immunopeptidomics, as well as about 50 samples profiled by ribosome sequencing to look at active translation at the open reading frame level, as well as 5 additional samples of healthy thymus, where we could perform HLA class 1 immunopeptidomics. For this, we wanted to take as stringent of an approach as possible, and so just because it's going to be really important, we take all of our peptides from our pancreas cancer immunopeptidomics, we map them back to their parent open reading frame. For any healthy tissue immunopeptidomics, we do the same thing, map the peptide back to the parent open reading frame. We cross-reference these, and if there's any evidence for translation of that open reading frame in even a single healthy tissue, we rule it out, and we say we can't be certain that this is tumor specific, so we're going to call that non-cancer restricted. Honestly, the first takeaway for us was that as a whole, we probably shouldn't be calling these neoantigens, at least as a class. For about 2/3, we could find clear evidence of translation of the parent open reading frame in one or more healthy tissues. And because we had class 1 immunopeptidomics from so many different organs and tissues, we could look even at the peptide level. For many of these, we could even find the exact peptide, really suggesting that these are not true neoepitopes, but rather that these are misannotations in terms of the annotated protein coding genome. But for the best that we couldn't, there were about 500, a little over 1/3 of these, that we could find zero evidence of translation in any of the healthy tissue samples profiled. And so we felt that these were likely tumor specific, but I still can't bring myself to say it, call it that, so we're going to call them cancer restricted, or CR, non-canonical peptides. One of the questions here was asking, well, is it just that we haven't profiled enough tissues yet? And if we had just profiled, a few hundred more healthy tissues, we'd really rule out all 500 or so. So we wanted to assess saturation. To do this, we took advantage of the fact that we had canonical peptides from our samples as well. The hypothesis here is that most canonical proteins should be translated in one or more healthy tissues. And so if we're nearing saturation, we should be able to rule those out as not cancer restricted. And in fact, that's just what we see. We can find that 99.5% of the canonical peptides are able to be ruled out as non-cancer restricted. The 0.5% that are left are highly enriched in known cancer testis antigens or oncofetal antigens, really suggesting to us that this 30% or so, or about 500, what we're terming cancer-restricted non-canonical peptides, are at least highly cancer-restricted. Again, can we say that they're never translated, processed, and presented in any healthy tissue under any circumstance? No. But again, highly cancer-restricted in these analyses. We then wanted to ask, well, are these just stochastic errors in translation? Is this just kind of the ribosome shifting and inappropriately? And so we wanted to compare the peptides that we were finding that were deemed to be cancer-restricted versus those that we found in healthy tissues. if these are just stochastic errors in translation, you would guess that certain classes, they would all be go down kind of in a graded fashion. And we don't see that at all. We actually see certain categories or certain biotypes profoundly enriched in the cancer-restricted fraction compared to the non-cancer restricted fraction. I'll call your attention to the INTORFs. These are these out-of-frame or alternative reading frame derived peptides. But it's not just the INTORFs. We can see, again, cancer-restricted non-canonical peptides are also enriched in link RNA translated peptides, as well as 3' UTR translated peptides. What we found was interesting is that many healthy tissues, again, we could find evidence of 5' UTR translation in healthy tissues, suggesting that those are likely misannotations in the protein coding genome and not actually true aberrant translation. One of the other things that pushed us towards saying that this is likely not just stochastic errors in translation, again, were the fact that so many of these were shared. I showed you before sharing at the open reading frame level. This is actually looking at shared at the peptide level. So if we're looking at sharing at the peptide level, again, these are HLA-1 bound peptides, so you have to look at kind of inpatient samples with the right HLA restriction element. Because these are Dana-Farber patients, we're very enriched in A0201, and so we can look at the number of cancer-restricted non-canonical peptides that are shared amongst multiple patients. And really exciting to us, about half of the A0201 restricted peptides were shared by 2 or more patients, with a large fraction actually shared by more than 70% of these A0201 individuals. Really, again, hinting at this is likely not just stochastic errors in translation. You'd have to make the same error a lot of times. But importantly, also suggesting that these are good therapeutic targets if we can develop TCRs or T cells to recognize them. The other piece that kind of pushed us to say, well, these could be very viable therapeutic targets is just how abundant they were. I showed you how abundant just these non-canonical peptides are, but this is looking at the cancer-restricted fraction as well. So the blue bars here are these cancer-restricted non-canonical peptides, how many per patient sample we're detecting, obviously trouncing the number of detected mutation-derived neoepitopes, but in many of the patient samples, about half, there are more empirically identifiable cancer-restricted non-canonical peptides detected by mass spec than the total number of exonic mutations present even at the DNA level, again, suggesting that this could really open up the antigen landscape for targeting in pancreas cancer if we can develop reagents to target them. And so for this, we wanted to start asking, well, what is the immunogenicity of these non-canonical peptides? And immunogenicity, I like to call it immunogenic potential for the following reason. So for this, we collaborated with Phil Greenberg's lab at Fred Hutch, adopted and then subsequently adapted approaches to do ex vivo T cell priming and expansion to look at the immunogenic potential of these peptides. Now, this is trying to mimic kind of ex vivo vaccination, but again, it's pretty artificial. But just to take you through it, we use HLA-matched healthy donor PBMCs or peripheral blood, isolate the monocytes, we differentiate these into dendritic cells using a defined cytokine cocktail, mature those DCs and then peptide pulse them with these candidate antigens, use these to prime autologous CD8 T cells, and these can then be boosted ex vivo a number of times prior to actually reading out immunogenicity using 2 metrics. One is the ability to stain with fluorescently conjugated HLA-1 tetramers, or the ability to secrete effector cytokines. Just to show you what some of this data looks like, this is an example of a translated 3' ORF or a 3' UTR-derived peptide, where you can see robust intracellular cytokine staining for both TNF-alpha as well as interferon gamma. When we did the tetramer staining, we needed to do this at scale, and so again, here we adopted and then adapted some approaches from Ton Schumacher to do fluorescent barcoding. So we can now stain up to 12 to 16 T cell lines at once using multiple different fluorescently conjugated HLA-1 tetramers, deconvolute these in flow space, and actually look for immunogenicity. This is a schematic, but this is what this actually looks like when we read out an aberrantly translated link RNA-derived peptide, and we can go back both that identify that we are seeing a tetramer-positive response. We use dual tetramers to confirm specificity, but then identify which T cell line actually had the response from this patient. What does this look like at scale? For these non-canonical peptides, to date, we've tested about 56 of these, and we were really excited. They were quite immunogenic. About a quarter of these showed quite robust immunogenic potential in this ex vivo priming assay. But what got us more excited about this was when we broke these non-canonical peptides up into those that were deemed to be cancer-restricted versus non-cancer-restricted, as one would hope, we found that the cancer-restricted fraction was much more enriched in positive immunogenic hits. Although I'll point out that we were able to break tolerance to 2 of the non-cancer-restricted peptides as well. We wanted to benchmark this and ask, well, how does this actually compare to the immunogenicity of mutations, kind of classic neoantigens? So for this, we went back and tested all of our empirically identified mutation-derived neoepitopes. But to expand that, we also computationally predicted mutation-derived neoantigens using multiple neoantigen prediction pipelines. And again, we could see quite robust immunogenicity here. For the computationally predicted neoantigens, we purposely varied this across a number of variables, across both predicted binding affinity for HLA class I, across a predicted agrotopicity, as well as tested a number of different categories, so both missense mutations, frameshift-derived mutations, as well as indels leading to these predicted mutation-derived neoepitopes. Lo and behold, mutations are quite immunogenic. Again, I'm a big still believer in mutations, actually. I love them. And so we were very happy to see this, but our kind of takeaway was that as a whole, the cancer-restricted non-canonical peptides were about as immunogenic, both by proportion and magnitude, as these mutation-derived neoepitopes. The biggest difference, of course, is the mutations. Only 1 out of the 10 immunogenic hits was actually detectable on the cell surface of these tumor cells, despite being present at the genomic level in these same organoids. In terms of the non-canonical peptides or these so-called cryptic antigens, we wanted to ask, well, are there certain biotypes that are enriched in immunogenicity? And the short answer is no. We can see kind of across these, multiple of these different categories leading to quite robust immunogenic hits. This is shown here. Again, no particular enrichment for any one biotype over another in terms of immunogenic versus non-immunogenic. So then we wanted to turn our attention to say, well, we've developed this translation-centric filtering pipeline. Well, is the inclusion of thymus immunopeptidomics actually helpful or not? And so we really wanted to stress test it here and say, well, was it helpful? And it turns out it has very good negative predictive value, as one would hope. Meaning if we could detect non-canonical or translation in healthy thymus, we could never break tolerance to that. The 2 non-cancer restricted peptides that we were able to theoretically break tolerance to, we did not detect them in the thymus. Again, low numbers here, but still fitting with this translation-centric filtering pipeline may help prioritize what antigens to target downstream. Then we wanted to actually develop, or try to develop, reagents to target these cryptic antigens. So for this, we had some T cells, but we actually wanted to identify the TCRs, the T cell receptors. And so for this, adapted that ex vivo priming and expansion platform. Now, instead of doing fluorescent barcoding, we're using DNA barcoded tetramers paired with the 10x single cell platform. So now we can read out both gene expression, paired TCR alpha beta, and antigen specificity because of the DNA barcode on the tetramer, and then look for the TCRs that are actually specific to these non-canonical peptides. So we were able to identify 17 of these cancer-restricted non-canonical peptides and their cognate T cell receptors using this approach, both A11 restricted and A0201 restricted across a number of different biotypes or categories. We were able to then take these and molecularly reconstruct a number of these and put them into lentiviruses, where we could then use CRISPR to knock out the endogenous TCR alpha beta from healthy donor CD8 T cells and create non-canonical antigen-specific TCR-T. We could just show you that the CRISPR knockout is very good, and we can knock out like 99% of the TCR, and that's great. But we can then redirect these using guide-resistant lentiviruses, encoding these TCRs to again, redirect antigen specificity. We can then take these, we can flow sort them, put them through a rapid expansion protocol, or REP, to generate tens to hundreds of millions of non-canonical antigen-specific TCR T cells for further functional testing. The first thing we wanted to do is start testing just biochemically. Are these all low avidity TCRs? Are these high avidity TCRs? And so we wanted to characterize this, and kind of the further left, meaning the high avidity, so meaning that these TCRs can trigger at a lower antigen density. These are the good TCRs, whereas the ones to the right require largely supraphysiologic levels of peptide to actually be triggered, and so we're going to deprioritize those ones. But actually, these three were actually in the low nanomolar to low picomolar avidities, and so we really prioritize these high avidity, non-canonical antigen-specific TCRs for further functional testing. So we took these, and then we created these non-canonical antigen-specific TCR-T to put them back in co-culture with patient-derived organoids, and now ask, really for the first time, can these actually recognize and kill tumor cells expressing endogenous levels of these cryptic antigens? Now, I probably wouldn't be telling you this if it didn't work, but it did. And so we could see clear evidence of recognition, both by triggering of surface 4-1BB or CD137 on these T cells. Again, when you put them in co-culture with the organoid, that we detected the peptide by immunopeptidomics, not at all if you put it with an HLA mismatched organoid. Not just 4-1BB, but we could also trigger effector cytokine secretion. This is interferon gamma. Now 3 different TCRs, the high avidity TCRs, we could see clear evidence of antigen-specific recognition. We could completely inhibit this just by sterically blocking HLA class 1 using functional blocking antibodies. And then we took that highest performing TCR, that TCR1, which recognizes this so-called what we call nu11 peptide. This is out of frame translation in the DDX47 gene. And we actually wanted to alanine scan this to look for potential cross-reactivity, meaning is this just a promiscuous TCR, or how specific is it? And it turns out it's exquisitely specific, meaning if we substitute any other amino acid at almost any other residue, we completely abrogate TCR recognition. Only position 6 was even mildly tolerated, and that shifted the functional avidity over 20,000-fold, suggesting it's a highly specific TCR that we've identified here. We further wanted to ask, well, can they recognize, but can they actually kill tumor cells? So for this, we had to develop this organoid TCR-T co-culture platform. We engineered a number of these patient-derived organoids now to express firefly luciferase, so we can read out viability in this system. When put in co-culture with these 3 high avidity non-canonical antigen-specific TCR-T cells, we can see quite exquisite cytotoxic activity, even at low effector to target ratios. And again, this could be completely inhibited by functionally blocking HLA class 1. We wanted to go a little bit further and not just use luciferase, but also engineer these to express different defined fluorophores so we could watch this happening in real time. So here we're able to grow these patient-derived organoids, image these on the IncuCyte, allow them, put them in with the specific TCR T cells, watch those T cells actually infiltrate here over on the right, and kill the cognate patient-derived organoids. So again, multiple lines of evidence really getting us excited about the functional potential here, but this is all in vitro or ex vivo. So then we wanted to put this kind of really stress the system, and we adopted some of these patient-derived organoids for in vivo growth using transplantation to immunocompromised animals. So we could allow these tumors to form and then run adoptive cell therapy preclinical trials using irrelevant TCR-T cells or our highest performing non-canonical antigen-specific TCR-T. And here again, we were able to show a quite robust delay in tumor progression. I want to be clear here that we did not eradicate tumors, although it's a very small number of TCRs tested to date. But again, we were happy as a proof of concept that these antigens could actually be recognized by these TCR-T cells, not just in vitro or ex vivo, but even in vivo, recognize and kill, really hinting that these could offer a viable therapeutic strategy once we identify the proper antigen, the proper TCRs. So for this part, let me just say, hopefully I've convinced you guys that these non-canonical peptides are quite abundant in the pancreatic cancer immunopeptidome, that a subset of these cryptic peptides or these non-canonical peptides are in fact cancer restricted. A subset of those cancer-restricted non-canonical peptides are actually quite robustly immunogenic, can be targeted by CD8 T cells. And then importantly, these non-canonical antigen-reactive TCR-T cells are truly capable of recognizing and then killing patient-derived organoids expressing endogenous levels of their peptides. And this both was shown ex vivo as well as in vivo. Now, this is one of these things where it kind of starts off as one project in the lab, and it kind of, in a good way, takes it into like 10 or 20. And so we're now active multiple other projects that we're following up on. Again, we're biochemically and functionally characterizing a suite of additional non-canonical antigen-reactive as well as mutation-reactive TCRs. We're now pushing this to ask, well, there's a number of clinically relevant perturbations in terms of chemotherapies as well as targeted therapies emerging in pancreas cancer. How do these impact non-canonical ORF translation or cryptic antigen presentation? We're building this, everything I've talked to you about today has been class 1 immunopeptidomics, building this in to look at class 2 immunopeptidomics in pancreas cancer, and then some ongoing work in terms of really trying to stress some of these TCRs or TCR-T for preclinical evaluation and hopefully in the future clinical evaluation. However, in terms of near-term clinical applications, we're actively working with Rishi Sarana, Kathy Wu, Patrick Ott, and many others to move this into the clinic actually as a pooled vaccine targeting these shared cancer-restricted non-canonical peptides in pancreas cancer, but may actually have implications in other tumor types as well. But I thought I'd spend just the last couple minutes on this, which is, I started off with this cancer immunity cycle and say, if we think about what steps are defective, then we can guide therapy. And really, all of the focus has been on trying to understand what antigens there actually are in these tumor cells and how can we overcome it. But even if we're able to develop vaccines or TCR-T, we still have to deal with this profoundly immune suppressive microenvironment of pancreas cancer that are going to exclude T cells or render these dysfunctional. And so my lab is now collaborating with Phil Greenberg's lab, who has really pioneered a number of these technologies, but kind of adopting and then iterating on these. Again, taking much of the discovery of novel PDAC antigens that we've been doing through immunopeptidomics, pairing that with TCR-T discovery, again, not just of these cryptic antigens, but also of mutation-derived neoantigens, and trying to figure out how these should be best paired together. Importantly, because cellular therapies, you can armor these, right? And so you can engineer these not just to use CRISPR, we can knock things out, like knock out PD-1, sure. But you can also create these immunomodulatory fusion proteins or so-called switch receptors, which are really elegant ways if we can take advantage of what we know about the tumor immune microenvironment, specifically in pancreas cancer, and turn negative or inhibitory signals, instead turn them into positive signals by fusing the intracellular domain of either 4-1BB, CD28, other co-stimulatory molecules. a number of other efforts to try to promote T cell persistence, T cell sustainability by actively engineering CD4 T cells along with these work that is still in process, but will hopefully be ready to talk about soon. So with that, I just want to thank the folks that actually did the work. Big group, and I tried to highlight the folks who did this throughout, but let me just call out a couple people. So really, this work was led by Zach Kolstad in my lab and Zach Ely, a former graduate student in Tyler Jax's lab, with tremendous effort from a number of people in my lab who are like sitting in a row back here. So Gurjan Gunadine, Sudarsana Adipali, Sila Wong, Marta Casarubios, Rafa Vandenbrink, Alex Blaine, Mike Wong, and many others. I mentioned kind of all of the immunopeptidomics was done by the Carr group at the Broad Institute, so really indebted to Jen Abelin and Steve Carr for making this possible. Again, they're kind of the world leaders in immunopeptidomics. Highlighted Phil Greenberg at Fred Hutch, and then again, super indebted to the many collaborators here at Dana-Farber in the Hale Center for Pancreatic Cancer Research. I highlight both Andy and Shri helped develop and characterize a number of the patient-derived organoids, as well as Eric Smith, who we've been collaborating with to do some of the functional testing. Last but not least, the funding sources that actually made this possible. So this was funded by Stand Up to Cancer, Pancreatic Cancer Action Network, the Hale Center, and Breakthrough Cancer. So thank you all very much.
Thank you. That was